Hello, good morning, everyone. We're going to spend the first 10 minutes perhaps to finish up lecture 6, and then we're going to start on lecture 7. So let's do a quick recap on where we are at so far. Last time we talked about the cell cycle. The cell cycle is basically the life of a cell from the point where it's just freshly formed, right, to the point where it divides again. Right? That the whole entire time is called the cell cycle. And the cell cycle, as you might remember, is separated into two key phases. Right? We have the interphase as well as the actual mitosis. And the cell spends, like most of the time, 90% of the time in interphase growing and preparing for the actual division, right? And we talk about how inside the interphase, there are three separate stages. G1 and G2 are checkpoints to see if the cell is ready for divide, right? So before you do the DNA replication in the S phase, you are going to have to check for the quality of the DNA. Are the telomeres too short? Do you have any kind of DNA damage or errors that needs to be repaired? Is the cell big enough? You do all these checking before you allow the cell to go ahead and copy the genetic material. Afterwards, you have to do another checkpoint. Like, did you copy everything? Did you make any mistake when you copy the DNA? If so, can we fix those mistakes, right? And only if all those things are good, then the cell is allowed to go on and do the mitosis, which of course has 4 phases to it, right? The prophase, metaphase, anaphase, and telophase. Then we talked about within the checkpoints, there are 2 kinds of proteins that are responsible for doing the checkpoints. We have things that accelerates, that speeds up the cell cycle, and we call those growth factors, right? And on the other hand, we have the growth inhibitors. Now, these are often proteins, and proteins, as you know, are encoded within the DNA. The instruction to make them are encoded within the DNA. And so the gene is a section of a DNA that gives you the instruction to make the protein. So for growth factors, the gene that makes it is the oncogene, right? The proto-oncogene. Proto-oncogene. And on the other hand, the gene that makes, oncogene, the gene that makes the growth inhibitor, that's the tumor suppressor gene. Okay, and we talked about how when there is a mutation in these genes, it could cause problems. So we can have a gain of function mutation in the proto-oncogene, and that changes it to an oncogene. Okay? And this gain of function mutation causes the green lights to be always on. And when the green lights are always on, the cell would keep on dividing and dividing despite being not supposed to. Okay? There could be, mistakes, and there could be errors. It just doesn't care. It just keeps on dividing and dividing. So this gain of function mutation is just one hit. Okay? Remember, I said you have 2 green lights, and if one of them mutates and is always on, that's enough to cause the cell to keep on dividing. On the other hand, the tumor suppressor gene has what we call like a loss of function mutation. And this one requires 2 hits. Okay. Loss of function for the tumor suppressor gene means one of the excuse me, one of the red light is not working, but that's okay. You still have a backup red light. Okay, it's only when both of them are not working, then you have nothing to stop the cell from progressing through the cell cycle, and then it divides and divide and divide. That's basically where we left off last time. Does anybody have any questions before I continue with the lecture? Okay. So once that happens, once the cell has accumulated enough of these mutations, then the cell will continue to divide in an uncontrolled manner, and that causes the formation of a tumor. The fancy word for a tumor is a neoplasm. Neoplasm. The neoplasm can be benign, okay, which is usually not dangerous and can be removed surgically in many cases without further complications. But if it's malignant means it is going to be cancerous, and it could continue to progress aggressively, and a lot of the cases we don't have an effective treatment for cancer, and it could lead to death. So how do scientists, how do people know whether it's a benign tumor or if it's a malignant tumor, right? What kind of guideline do they use to determine one versus the other? And one of the first things that people do when they discover a tumor is to do a biopsy, right? Take a sample of the tumor, and then to determine whether it's the benign version or the malignant version. Now, before we talk about the characteristics of cancer that allows doctors to know whether it's malignant or benign, here's a quick simple diagram that explains the progression of cancer. It's in a very simple terms, right? So this is a normal cell, and then there is going to be some kind of mutation. And the mutation, like I said, could be spontaneous, something that just happens just because of bad luck, okay? Just by chance, when your cell divides, when they copy the DNA, they made a mistake, and that mistake was never caught or corrected. There's a spontaneous mutation. It could happen to any one of us, okay? It's just unlucky, right? But the other one is induced, right? It's because of exposure to carcinogens, right? Things that causes DNA to mutate. And there's no shortage of carcinogen in this world, right? For example, cigarettes, right? There are a lot of things in there that could cause DNA to mutate. And once you have accumulated enough mutation, right, the cell starts to kind of continue to get more and more mutation, and this is how it works. Okay, basically, imagine having a tumor suppressor gene that encodes for a protein that helps you fix DNA damage. Okay, the job of the protein is to help you fix DNA damage. If that protein is not working, then what's going to happen? You're just going to end up having more and more and more DNA damage, right? So usually some mutations are like worse than others, right? Some mutations will cause like an avalanche, like a domino effect of additional mutations. And when there are enough mutations, it would just cause the cell to become very, very aggressive, and then it keeps on growing and growing and growing. And sometimes, for some types of cancer, the cell is able to detach from a tumor, travel through the bloodstream or in your lymph nodes, and it goes to other parts of the body and spread. And ultimately, death is imminent for many forms of cancers. Typically, when you have a cell that detaches from the body, the cell doesn't live. But in the case of cancer, they have mutated in a way that allows them to survive, even though they are detached from the original tissue. So back to the point I was saying, right? Like how does what characteristics, what parameter do a physician use to distinguish whether the tumor is benign? Benign again is a good outcome, the harmless version, or if it's malignant, which is the cancerous version. And so there are 6 characteristics associated with cancers, and they're all summarized here. And then I'm gonna go and explain each one of them in more details. So hallmarks of cancers, characteristics of cancer. First one is self-sufficiency in growth signal. Okay, they do not need signals to tell them to divide. They have mutated in a way that they have their own green lights, right? Remember the oncogene? They got their own green light. They are ignoring regular signal. They don't even need to pay attention to that. They have their own self-sufficient signal, and that causes them to continuously to divide. Not only that, their red lights are not working either. So they are insensitive to anti-growth signal. Okay, so you don't have to tell them to go and they will just divide on their own. Worse, when you tell them to stop, they don't listen. They just keep on dividing, even though the signals are telling them to stop. It's insensitive to these inhibition signals. So basically, these two things combined together causes the tumor to form, right? The neoplasm that I was telling you about. So those are 2 characteristics. Next, they can evade apoptosis. Right? One of the checkpoints outcome is to tell the cell to die when they are not in good shape or form, right? If the DNA has sustained too much damage, and you cannot fix it, or if the cell is behaving in any kind of strange way, you are supposed to trigger apoptosis. But in the case of cancer, they can evade apoptosis. They block the apoptosis pathway, right? You are not able to kill them off. They essentially become immortal. Now, with a lot of cells forming, it needs additional nutrients and a lot of supply. So they actually can create their own network of blood vessels. That's what angiogenesis means. Genesis is to create, angio is blood vessels. Okay, so the sustaining angiogenesis means they are able to create their own network of blood vessels to supply the tumor. And often it basically sucks nutrients away from healthy cells. Okay, so not dying, have a direct line of supply for energy to support the rapid growth of the cell. And they also have limitless replicative potential. Okay, we talk about how in normal cells, because of the lack of telomerase, the enzyme, the telomeres will get shorter and shorter and shorter, and eventually you're not allowed to divide anymore, right? But in the case of cancer cells, they found a way to turn on telomerase. They express telomerase when it's not supposed to. And that allows them to reset the telomeres every time they divide, and they no longer become senescent. Senescent is when the telomeres are critically short and the cell is not permitted to divide anymore. It becomes old, right? It becomes senescent. But that's not a problem with cancer. And again, that's why they could keep on divide and divide. And finally, in advanced stages of cancer, they are able to invade other tissue and they're able to metastasize, to spread to other parts of the body. Okay, so those are the 6 hallmarks of cancer. Right? They have their own growth signal. They ignore inhibition signal. Right? They can evade apoptosis. They have their own source of blood vessels. They can keep on dividing and dividing and dividing, and then they could invade and spread. All right? That's it. Any questions? All right, let's go through some slides here. Penetrance is best described as, I mentioned penetrance last time. It basically is the concept that tells you how likely will the person end up having a disease if they have the mutated gene, right? So the answer is C, the percentage of individuals in the population expressing a gene that they carry. Okay. What is a proto-oncogene? A proto-oncogene, a gene involved in promoting normal cell growth. That's the correct answer. A proto-oncogene is the normal version of the green light, allows the cell to grow. A gene involving in slowing down the cell cycle, this would be a tumor suppressor gene. And a gene that causes cancer, well, it wouldn't be the proto-oncogene. It would be like the oncogene, right? The mutated version of it. And for proto-oncogene, I wrote it down for you earlier, it's not a loss of function that increased cancer risk, is a gain of function, right? Remember, the proto-oncogene are green lights. If the green light does not work, like if the green light does not turn on, that's not a problem. That's not going to cause the cell to keep on dividing. It might cause some other problems, but it certainly will not cause the cell to keep on dividing, right? If the green light is not turning on, worst case is the cell don't divide. Right? But if you have a gain of function mutation, that means the green light is always on, and then it will always be dividing and dividing. Okay, maybe we try this one. Right, let me know what you think. Okay, I'll launch the poll. Anyone else? Okay. The correct answer is that it does not accelerate the cell cycle. Okay. Tumor suppressor gene are the red lights. They stop the cell from dividing. Okay, and it has to do with fixing up problems. Okay, so it's responsible for DNA repair. They could kill the cell, right? Killing the cell is going to suppress the tumor, and check for DNA damage. It does not accelerate the cell cycle. That would be the job of a proto oncogene. Okay. And I think that is the last one for the lecture. Okay. That is lecture 6. Then we're going to move on to lecture 7. When is our quiz? Do you guys remember? I think it's next week, right? You just double check on that really quickly. This is week number 8 or 9. Honestly, it's all blended together for me. Today is the 7th, right? Yep. So this is week 9, and the quiz is going to be next Tuesday. Okay, next Tuesday. And we will also have a lab next week, the microscopy lab. So I will tell you more about that later. But now let's move on to lecture 7. So lecture 7 focuses on protein synthesis. Right? We know how the information, the instruction to make the protein is encoded in the DNA. And we know that to go from DNA, we need to first make a copy in the form of RNA, and then we're going to use that instruction to make the protein. Okay, so this first part here is called transcription, right? From DNA to the RNA is transcription, which happens in the nucleus, and then from RNA to protein is called translation. Okay, so that is going to be the main focus. Do you want to work on study? What do you mean, Mozaide?
Last you say print out study guides
Yes,
and
we're gonna work on the study guide for lecture 7, right?
yeah.
Yes. So yeah, we'll be doing some questions from there for sure.
Okay, today you want to
Today,
do?
yeah, today.
Yeah. Okay.
Thank you.
Thank you.
Madison, when will the answer sheet for the study guide be posted? Are you talking about lecture 6 or 7? Six is already up, I believe. I posted last night. Please double check that, and if it's not working, let me know. Thank you. All right, so transcription from DNA to RNA. So there are a couple of terms I would like to, refresh with you, or like some of you might, learn this from the first time. I don't know, but there are some key terms that we must go through. Right. So when we say the genome of an organism, we are talking about all of the genetic information. So the human genome is all of the chromosomes, all 46 of them, number 1 all the way to number 23 pairs. Okay, that's all the genome. Think of the genome as a textbook. You don't think it's there? Okay, let me see. 201, and then content, study guide answer, number 6, right? Study guide answer. It should be there. Okay, just double check again. All right, so genome. Genome is all the genetic material. Think of it as your entire textbook. Okay. A gene is a section on the chromosome that contains instruction to make a protein. All right. So in this textbook, which represent the genome, there are 23 chapters. Right? So each chapter would represent the chromosome number. Okay, so chapter 1, chromosome 1, chapter 2, chromosome 2, so on and so forth. Okay, and then the gene would be like a section within the chapter. When you read a textbook, chapter 1 talks about the scientific method, and then section 1.1 talks about hypothesis, for example, right? So that section, that 1.1 and then 1.2, 1.3 within the chapter, that's the gene. Okay, this is a location, is a segment of the DNA that gives you the instruction to make the protein. So gene A here tells you how to make protein A. Gene B tells you how to make protein B, and so on and so forth. And the location at which you find a gene is the locus. So that would be like the page number. Right? So like you would find section 2.3 on page 163. Okay? The locus is the page number. All right. Now remember, we inherit 2 copies of each chromosome, one from our mom, one from our dad, right? And so that's like having 2 textbooks. Right? One textbook is maybe English version. The other is like maybe French version. Okay, they have the same information but slightly different way of expressing it. Right. So if you want to find the location of gene B in one book, like I said, page 163, you will find the same information on page 163. But again, because it's a different language, it's kind of expressed a little bit differently, right? So, you inherit gene B from say mom, and then she had blue eyes, then gene B is blue eyes. But if you inherited gene B from your dad, and he has brown eyes, then it's like a different version of the gene, right? Okay. So you guys understand this? Genome is all of it. Gene is a section on the chromosome that allows you to make a certain protein. And where do you find that gene? That would be the locus. Now, the chromosome 1 from mom and the chromosome 1 from dad, they are called homologous chromosomes. All right. So chromosome 1 is homologous to chromosome 1. Chromosome 2 is homologous to chromosome 2. 3 is homologous to 3, and so on and so forth. These homologous chromosomes are the same length, same size, and they have the same order of genetic information. Okay. Like I said, if you find gene A here on one, you're going to find gene A at the same place on the other one. Okay? And again, the gene actually, I keep on saying the gene gives you the instruction to make the proteins, right? But those proteins will cause you to have different appearances. Okay, so for example, we could have a gene here for eye color. I'm making this up, okay, eye color. Then you would find eye color on the other side as well. You might have one for blood type. You might have one for the kind of hairline that you have. You might have one for like the type of earlobes you have. Are they attached or are they detached, right? So you will find it in the same order on the homologous chromosome, the other homologous chromosome, but the version might be different. It could be the same, right? Maybe both your parents have the same eye color. That's why they're both like big A here. But if one of them has a type O blood and then the other has type B blood, then it's different version. Okay. And here hairline, one might have the straight hairline like this, the other might have a bit of a widow peak, and that's different. And earlobe maybe it's the same. So same order of genetic information, but the version of the gene might be different. And a different version of a gene, we say allele. Okay, allele. So
can you just, sorry,
go ahead.
Can you talk about the homologous chromosome one more time? I'm a bit confused.
Sure. Okay. But let me finish my sentence first. So allele would be, if you think of eye color, then there are different alleles for it, right? There are blue eyes, brown eyes, green eyes, right? These are different versions. And so we call them alleles. Okay. So again, homologous chromosome, right? We have 23 pairs of chromosomes. So this is chromosome 1 from mom, chromosome 1 from dad, like your mom, your dad, right? And then this is chromosome 2 from mom, chromosome 2 from dad. So 1 and 1 here, these are homologous. Okay, they are a pair. They are like think of, you have 23 pairs of shoes, okay? Your red sneaker is your left red sneaker is homologous to your right red sneakers and nothing else, right? So each pair of shoes is essentially the homologous chromosome. Does that help a little bit, the analogy?
Yeah.
Okay. So same size, same length, right? Same order of genetic information, but the version of it might be different. We're gonna use these terms quite a bit. So anyone else have any other questions? Like if you're not clear, I can try to explain again in a different way.
Sorry, they're like the same thing, but they might have a bit different genetic information in them.
They have the same order of genetic information, but then the
Okay.
version of it might be different.
Okay.
The version of it, the allele might be different. Okay. Another analogy that might help you is this. I mean, it might not help. I don't know. Like remember when we used to have CDs, right? Like to listen to song, you can think of a CD rack. Okay, so like you have two CD racks. Each rack represent a chromosome. Okay, so you will have the slots to put the CDs in, right? Okay, so like over here, right, you would put an artist in there, like I don't know, like put Adele in there or something, and then over here you would have to put the same artist, but then it could be a different album, right? So like the different album is like the different alleles, and then on the next slot you would put another artist, okay, Beyonce, right? But then you will have to put Beyonce here as well, right? So the order of the artist would be the same. That's like saying means the order of the gene are the same, but then the version might be different. You don't have to put the same CDs in there. You can put like different CDs, different albums, right, by the same artist. So keep that in mind as we go on. Now, this is the central dogma of biology, which we've learned before, and you know it's an overview of how you take DNA, and then you're gonna make a copy of it so that it forms the pre-mRNA. Gonna talk more about that later. It turns out it doesn't just change to the RNA immediately. You know, there is a pre-version of it, and you have to modify a little bit before it becomes the actual messenger RNA. And then that's gonna exit the nucleus through the pores, and it is in the cytoplasm that you will use the ribosome to decode the RNA to make the polypeptide, the protein. Okay, so that's the overview, right? There's just one additional new piece of information here. There is this pre-mRNA that we didn't talk about before, but it's just like an additional step that I will give you more information on in a bit. All right, why do we have to do transcription? Let's just refresh our memory on the reasons. DNA is very, very big, right? Like you have a lot of it all in the nucleus, and it won't even fit through the nuclear pore, even if we wanted to take them out. The only time we want to take the DNA out is during mitosis, right? Well, and we learned that last class. The nuclear envelope is going to dissolve, and then the DNA is going to come out, right? That's the only way for it to come out. Otherwise, it's too big to leave the nucleus. And we don't want the DNA to come out and you know stay in the cytoplasm. There are a lot of like enzymes there that might you know accidentally damage and degrade the DNA. So we don't want to do that. Instead, when we need the instruction from the DNA, we just find the gene that we're looking for, and then we copy that section. Okay, and that's useful because we do not turn on all the genes all the time. Right? We have thousands of genes in a cell. We don't need to make all of them. We don't need to convert all of them into proteins all the time for all the cells, right? It depends what kind of cells you are. Take telomerase, for example. I only need to turn that on in germ cells. Every other cell, we leave it alone. We're not gonna convert it into proteins, right? And that's the same for many other genes. Okay, in the liver, you might need some proteins to break down toxins, so you turn those genes on. But we don't need those genes to be turned on in the muscles. Okay, so all the cells have the same set of genes, but which one you convert into protein, which one you turned on, and which one you leave off, that's going to be dependent on the kind of cell that we are looking at. Okay? So, base pairing rule, we know A goes with T and G goes with C. But in terms of DNA and RNA, they could also pair as well. I mean, it's not a new concept, right? When we talk about the primer, we already know instead of the thymine pairing with the A, we have the uracil instead. Okay. Now this part is a little bit complicated. I don't know why, but like people usually gets really confused. So I'll try to go slow, and it's important you let me know when you're confused. ask questions so I can help you, understand better. We have 2 strands of DNA, right? 2 strands of DNA. Let's say we have, I'm just making this up. You don't really have to copy it. Okay. And then it's, it's really, really long. And then the other side would be like TAGGCGTA like that. Okay, 2 strands of DNA. Anti-parallel to each other, complementary to each other. So which one contains the instruction to make the protein? Right? Is it the top one? Is it the bottom one? Like, we don't know. Okay, but the key thing is only one of them has the instruction to make the proteins. Okay, only one strand, one of the strands has the instruction to make the protein. Okay, and you cannot think of it as the top one or the bottom one. Top and bottom is meaningless, right? Inside a cell, like it's just top and bottom because of the way I've written it, right? Like in the cell, it could be upside down, it could be like turned sideways, like you don't know which one. Okay, but the key thing to know is only one of the 2 strands is the one that actually contains the instruction to make the protein, and that strand is called the coding strand. Okay, that's the one that has the DNA code, the instruction that tells you which amino acids to put together to form the protein. That's the coding strand. And like I said, you don't know which one it is. I have to tell you on the test. Like if I give you 2 strands, I'll have to tell you, okay, like the top one is the coding strand, or I would have to tell you, oh, the bottom one is the coding strand. There's no way for you to tell just by looking at it. Okay. So what's the other strand doing? The other strand that is not the coding, the other strand that is just kind of there as a complementary strand, that's the template strand. That's the strand that will be used to make a copy in the form of RNA. You understand this? So 2 strands. 1 strand has the instruction. 1 strand is used to make a copy. Coding, template. Any question?
So one has like all the stuff you need, then the other one needs to copy all the stuff you need so it can go make more stuff.
Yeah, essentially. Yep.
Okay.
That sounds about right.
Okay.
Okay. So here it is. A question that you will have to know how to solve. Given the template strand, find the coding strand and the messenger RNA. Okay, so this is the template. I'm going to write it down here. Template. Template strand. Okay, so let's write the coding strand. Now it's easy to write the coding when you're given the template, and vice versa, because the template strand and the coding strand are complementary to each other, right? They follow the base pairing rule. So if you see T on the template strand, you know it's gonna be T. Sorry, I said that wrong. I said the answer instead of the question. If you see A on the template strand, you know it is going to be T on the coding. So, you should do this with me. You can say it out loud in your own room, okay? So T, T, G, A, C, A, G, C, T, G, A, T. Okay, these are just the double helix. They are complementary. Okay? Complementary. They follow the base pairing rule. That is very straightforward, right? Now, the messenger RNA. Messenger RNA. We have to make a copy of the DNA so that we can use it to make the protein in the cytoplasm. So the messenger RNA, tell me, should the messenger RNA be the same as the coding strand, or should it be the same as the template strand? What do you think?
Template strand?
Okay, anyone else? I am trying to make a copy of the instruction to make the protein. So should that be the same as the template strand or the coding strand? What about the rest of you? Do you guys agree, or you have a different opinion? Okay, Sarah said coding. Okay, I'm gonna do a quick poll. Please do the poll. Okay, I don't know why you guys are not participating with the poll. If you think the messenger RNA has to be same as the template, choose A. If you think it has to be same as coding, please choose B. Remember, we're trying to copy the instruction to make the proteins in the cytoplasm. So which one should it be identical to? Okay, I think I have a good outcome here. 10 of you answered. And the correct answer is coding strand. Look, if the coding strand contains the instruction to make the protein, then of course your mRNA wants to be the same as the coding strand, right? The template doesn't have any information, right? So the mRNA is going to be exactly the same as the coding strand, with one key exception. There is no T in RNA, so you have to change it to U. Everything else is going to be the same.
I have a quick question.
Yep.
Because I said template strand because on the previous slide it says the strand of DNA that is used as a template to make the RNA copy, the mRNA copy. So I'm a bit confused why it would be the coding strand.
Keep that thought. It's gonna make more sense in a bit. I have to show you a picture of how it works in a bit, and then it will work. But valid point. I understand why people choose template strand now. Okay, but hold on. Okay, just hold that thought. It will be answered in a couple of slides. Are we okay? Other than that little point that Madison just bring up, again, good point. Do you understand why it has to be the same? Okay, because look, this is the code. The code is the instruction. So of course you're gonna copy the code, right? Okay. Let's try another one. This one, create mRNA from the given template strand. Okay, so this is the template strand. And how do you do the mRNA? Well, let's go back here and take a closer look here. The template strand and the mRNA, they are actually complementary as well. Complementary. It follows the base pairing rule. A pairs with U and C pairs with G. So if you are given the template strand and you want to make the messenger RNA, right? This is template strand. All you have to do is pair up the nucleotides. So A is gonna be pairing with U, T is gonna pair with A, C will pair with G, and so on and so forth. Okay. Follow base pairing rule for DNA/RNA. Okay. So A pairs with U, C pairs with G. That's when you are given the template strand. Okay. What if you are given the coding strand? What if you're given the coding strand?
Can you please go back for a quick second?
Sure, sorry. Let me know when you're good to go.
So sorry, when it has T, you put A there, but when it's A on the top one, you put U on the bottom one.
That's right, because A pairs with T, right? But
Mhm.
then there are no T in RNA. That's why we have to use the uracil instead, right?
Okay.
Okay. What about coding? If you're given the coding strand, right, what would be the messenger RNA? If you have the coding strand and you want a messenger RNA, it's very straightforward. You just copy everything the way it is. But because we're talking about RNA, we have to use uracil instead of thymine. But other than that, everything stays the same. Okay. Simply replace T with U. Remember, coding strand and mRNA contain the exact same information. Okay? We want it to be the same. It's the instruction to make the protein. Of course, I'm going to copy everything. Right? But the problem is I don't have thymine in RNA. That's why I have to make the switch to uracil. Now, if you're confused as to the rules, this next slide will summarize it for you. Coding strand and RNA are identical, except uracil replaces the thymine. And then the template strand and messenger RNA, they are complementary to each other, where ATCG pairs with UAGC. Okay, so cheat sheet. One of the things to put on your cheat sheet. Coding. Okay, if coding is ATCG, again, there are only 4 possible letters, right? Then the template is going to be TAGC. And what about mRNA? What is mRNA going to be? Someone tell me. Okay, Kiana said UAGC. Okay. Anyone else?
AUCG.
That's right, AUCG, because it's same as the coding, right? Same as the coding. Okay, not the same as the template. The template doesn't have the instruction. Okay, the template doesn't have the instruction. So you don't want to be copying the template. If you end up having the template, you're not going to be able to make the protein because once again, the template does not have the instruction. Okay, maybe I show you a picture of like what it looks like here. I'm just going to skip ahead and show you here to help you make sense of it a little bit more, okay? How should I do this? You know what? I can't skip too many things, otherwise it wouldn't make sense. Let's skip the poll questions and let me try to give you a little bit more context here. Okay, so we are talking about the process of transcription. Right? Transcription is essentially DNA to messenger RNA, right? Okay. Like we're not converting the DNA into messenger RNA. We're making a copy of the DNA in the form of RNA. So the enzyme that does this is RNA polymerase. Not surprising, because instead of making DNA, we're making RNA this time. That's why it is RNA polymerase. Okay. The polymerase, the RNA polymerase, will pull apart the DNA, and then it will use one of the strand as a template to build the messenger RNA. Think back to DNA replication. When you build a new DNA strand, you have to use the original strand to figure out what nucleotide to put, right? So if you see an A, you know to put a T. If you see a C, you know to put a G, right? Same principle here. You have to use one of the strands as a template to build the RNA strand. At the end, you get something called pre-mRNA. It's not quite ready to go out yet into the cytoplasm. We got to fix it up a little bit. We got to process it a little bit before it can leave the nucleus. So there are 3 stages here for transcription: initiation, elongation, and termination. To put it simply, you need to know where to start. That's the initiation. You need to actually build the messenger RNA. That's the elongation. And then termination is, you got to know when to stop. Okay, so initiation is like where to start. And this is termination is like where to end. And this is like actually building. Right? Building the RNA. Okay. Now, a gene is really, really long. It has a lot of ACTGs in it. How does your body know, where is the beginning? Where's the beginning? Right? I mean, imagine this. Imagine having a textbook again. Imagine having a textbook, but they take all the pictures away and then they take away all the space. It's just text. It's just letters. Okay, all the letters, alphabets. Okay, and no space at all. Okay, from the first page all the way to the last page. Just text with no space, no picture, no nothing. Then it's very difficult to tell where the beginning is and where the end is, right? Like where does one chapter end and where does the next one begin? Like, I mean, if you read it, you probably will be able to have some idea and figure it out, but it's really hard, right? Okay, so that's why in an actual textbook, we have these like, big pictures and then big titles, like, chapter 2 that tells you the next chapter starts on this page. So similarly, in a gene, we have a section that tells you the beginning of the gene, like where does it start? We call that the promoter. The promoter. A promoter marks the beginning of a gene. This is where you need to start copying if you want to copy this gene, if you want to turn this gene on. And the promoters are rich in A and T's. We know that we know the reason for that. It's because there are only 2 hydrogen bonds holding A's and T's together. So it's easier to pull apart the DNA at the promoter. Right? Whereas if it has a lot of C and G, it takes more energy to do it, right? So promoter is AT rich, and the RNA polymerase will bind to the promoter. It will unwind the DNA, and that is the starting point. This is where it's going to start copying. The elongation process essentially looks like this, okay? The RNA polymerase will unzip the DNA, it will build the RNA, it will move forward. As it moves forward, it will unzip more DNA ahead, but it will rezip the DNA behind. It's gonna close it up, right? So as it's unzipping more at the front, it's gonna close up the DNA behind, right? So only a small bubble exists at a single time. Now let's take a closer look at my diagram here. Right? This whole grayish color thing, that's the RNA polymerase. And here, the template strand is being used, right? You see the template strand is being used to construct, to create the RNA. Because you can only build using base pairing rule, right? So the RNA polymerase see that, okay, there is an A here on the template DNA. I'm gonna have to put a U there. There's a T, I'm gonna put an A. There's a T, I'm gonna put an A. G, I'm gonna put a C. So it's just following the base pairing rule to build the RNA. Now, if you compare the RNA, look, it contains the same information as the coding strand. You see the A here, T is replaced by U, C is C, C is C, A and A. Essentially, when this is done, the RNA that you've created will be identical, contains the same information as the coding strand. Okay, but you couldn't use the coding strand to build, right? If you use the coding strand to build, then you end up having like the opposite of what you needed, right? A loose analogy for this is like this. If you think about minting a coin, okay, like the mold that you use, to pour the metal in again, right, to make the coin, the mold has to be a mirror image of what you want to get in the end, right? Like if it's not a mirror image, then your coin is gonna come out looking like backwards, right? So in this case, it's the same thing. The template is not the actual code, is the complementary version of it, right? That's why the template strand, we said we use the template strand to build it, but then the information in the RNA is same as the coding. Does that make any sense here? I don't know if I'm making sense. Do you guys have any questions? No questions? Are you guys overwhelmed by by all my? Can I repeat that, please? Okay. I guess the easiest way to do this is just to draw it out, right? To see it. I think maybe that's the way to go. Okay, so let's say we have the little bubble here. Okay. So again, we just call the bottom the template strand. The template strand. Okay, and then this is the coding strand. Okay. First of all, you have to understand this. When you build DNA, or even when you build like when you build DNA or when you build RNA, because of the way the enzyme works, the enzyme cannot do this. The enzyme cannot see there's an A, I'm gonna put an A. You see a T, I'm gonna put a T. See a G, I put a G. It doesn't work like that. All the enzymes, the polymerase that we've seen, they cannot copy exactly as it is. The only way for them to build anything new is to follow the base pairing rule. So if it sees an A, it has to put a T if you're building DNA, right? If you see an A, a T, you have to put an A, so on and so forth. That's the only way the polymerase can work. It can only do base pairing rule. Are we clear on that? Okay. They can only do base pairing rule. So let's say we have the template here again. A, T, C, G. G, C, C, A, so on and so forth. Now, there are two strands of DNA here, right? So, of course, if this is A, on the other side, it must be a T. Dot, 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 dot. So the coding strand is the one that has the instruction to make the proteins. I want my final RNA to look like the coding strand. Okay. For that reason, when I make the RNA, I cannot build it based on the coding strand. If I did, this is what will happen. If you use the coding strand to build the mRNA, right, then it will look like this. It will look like A, U, G, 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 C, C, A. If you build your RNA strand using the coding strand, you would end up having something that looks nothing like the coding strand. The instruction is all backwards, if you would. Okay? Do you understand that? If I use the coding strand to build my RNA, the information on my RNA is completely opposite to what I need to make the protein. Okay? Because when you build strands, everything is flipped, right? Everything is according to the base pairing rule. So that's why we're not going to do this. Instead, we have to use the template strand to build our RNA. Right? So A is going to pair with T normally, but we don't have T. We have to use uracil for RNA, and then so on and so forth. Then my RNA here, my RNA is going to be the same information as the coding. That's why we need the template strand to build the RNA. Oh, thank you. What about the rest of you? Do you guys understand what I'm saying? And it's okay if you don't. Like, I mean, like this is hard to explain on a virtual environment. Yeah. Okay. So what's gonna happen next? Well, let me see what's gonna happen next. I gotta go back to the slide here. Okay, when are we gonna stop? Well, the beginning of the gene is called the promoter, right? So what about the end of the gene? The end of the gene is called the terminator sequence. Terminator sequence, right? Like the movie. Terminator stops the whole thing. So promoter starts, terminator ends, and then the RNA polymerase falls off, and then you have your pre-messenger RNA. Okay, pre-messenger RNA. That's it. Now let's go back to the poll questions for a bit here. All right. Please choose the answer here, and I'll launch the poll. Please participate. That's the only way for me to see if you're with me. Now on the test, if you see a question like this, don't even look at the answers. Okay, just solve the problem yourself, and then see which one matches your answer. Okay, otherwise you get like confused. Okay, I'm gonna launch the poll. Hopefully it's not gonna block the screen.
So questions like this will be on the test, is what you're saying?
Absolutely.
Okay.
Yep. Anyone else? I have 8 responses. Where are the rest of you? 7 more. Anyone else? Now, where is the cheat sheet? Oh, it's gone now. Okay, but I wrote down the cheat sheet for you, right? If you're confused on the test, look at the cheat sheet. I mean, I strongly advise you understand this, but then if you just can't wrap your head around it, then you need the cheat sheet, which is gonna be like this again. Okay, I'm gonna close the poll. Okay, I'll show the results. But look at the cheat sheet here. Remember, there are only 4 possibilities. So A, C, T, G here. And then the template would be complementary. I think this is the different order that I wrote it before, but it doesn't matter. It's the same 4 letters, huh? And then the messenger RNA. Messenger RNA has to be same as which one? Same as coding or same as template?
Coding.
Coding, that's
Coding.
right. So I'm gonna go ACUG, right? So write this down, stick it somewhere on the wall, okay? So now you see the question, template strand. Okay, so that means I'm given template strand and I have to find mRNA. What's the relationship between template and RNA? They are complementary, right? Complementary. So that means I have to use AAU, because look here, A becomes U, right? And then T becomes A, right? GCU, UGA. That's the answer. Okay. I hope that makes sense. Let me see what else is there. Lecture 7. Okay, we're gonna get to that later on after the break. But let's do another quick poll question here. Different version of a gene is called. I'll just give you a moment to think about it, and I'll tell you the answer. Okay. Anyone want to tell me the answer before I review it? Different. Yep.
The homologous pair. Oh no, it's the Elia.
Okay, yeah, so a few people say D, some say C. Different version of a gene is actually allele, okay? And the alleles will be found on the homologous pair, right? Like chromosome 1, right? Eye color, they could be different version, and then the different versions is called the allele, okay? Allele. Homologous chromosome, again, homologous pair is like chromosome 1 and 1, right? 4 is homologous to 4. 16, chromosome 16 is homologous to chromosome 16. Maybe I show you a picture to make this a little bit more relatable. There is a karyotype. Karyotype shows you all the chromosomes. Okay, how do I make this full screen? There we go. So look, this is chromosome 1 and chromosome 1. This one is curled up, but you know, if you line them up, it's the same length. So that's your homologous chromosome, 1 and 1. 2 and 2 are homologous. 3 and 3 are homologous, so on and so forth, right? You see these dark bands here? These dark bands and light bands, that's where the genes are. Right? So like over here, my mouse, there's a gene right there, and you will find the exact same gene on the homologous chromosome, on the other chromosome. But again, the version of the gene might be different depending on who your parents are, right? I keep on saying, if the gene is eye color, they could both be like brown eyes, or you know, one could be blue eyes and the other is brown eyes. They don't have to be different. They could be the same. But the version of the gene that you get is called allele. Okay, that was a lot of information. We should definitely take a break. We'll continue at 9:45. If you haven't, download the study guide or print it out yet, you absolutely need to do that. Because we'll take up some problems after the break. Okay, see you in a bit. All right, let's keep going. We've done all this already. So where are we? We just finished making the messenger RNA in the process of transcription. But like I said, the end product of transcription is not the final ready messenger RNA. Instead, we have this thing called the pre-mRNA. There should be an M in there. mRNA. Okay. And we need to modify this pre-mRNA to make it more stable in the cytoplasm. Otherwise, it's just going to break down very quickly, and we won't be able to use it for very long to make the protein that we need. And it turns out not all the DNA that you copy are coding information. Some of it is like filler DNA that we need to remove, and that's what we're going to be doing in post transcriptional modifications. Okay, so there are three things that needs to be done. Three things. Number one, we need to add a 5' cap, like you like putting a cap on a bottle, that kind of cap. Okay, this one is made of a guanine, a G. And then we're gonna add a poly A tail on the 3' end, and then we're gonna do something called RNA splicing. So let's take a look at the first two first. Okay, addition of a 5' cap and a poly A tail. So these two things, when you add the cap, which again is a guanine, when you add the cap and you add the tail, the poly A tail, which is just a bunch of A's, adenines, it stabilizes the RNA in the cytoplasm. And it turns out the more A's you add, the longer it will last in the cytoplasm. And that adds an additional level of control. The 5' cap is made of U. Oh no, the 5' cap is made of G. G is in like the guanine. Okay, G right here. Okay. So the poly A tail, the more A's you have, the longer it will last in the cytoplasm, and that's important for additional regulation. Think of a protein that you need to make constantly. Okay. So if I need this protein all the time, it makes sense for us to have the mRNA to have a very long poly A tail. So it's going to stay in the cytoplasm for a longer time, and we don't have to keep on doing transcription to make more of the RNA. Sometimes we have proteins that we just need a little bit of, and and that's it after that. In that case, you don't need to have a very long poly A tail. Okay, we don't need the mRNA to be hanging around for too long. We just make a bit of the protein that we need, and then that's it. Right? We no longer need it. We can let the RNA break down and degrade. Okay? So the caps and the tails stabilizes the RNA, and also the longer the tail, the longer it will last in the cytoplasm. Now, if we zoom in to the actual messenger RNA, it turns out there are alternating, there are regions that are the coding region, and then there are some non-coding region. Non-coding, you can think of them as like junk DNA. Okay, junk DNA. They don't really do anything. That's not entirely true, but whatever, it's okay. It's okay. We'll just think of it like that. So these junk DNA are called introns. Okay, and we don't need them. Whereas exons are the coding region. They are the part that is being expressed, and we want to keep them. So if you take a look here, we have a bunch of exons here, which we're gonna keep, and we're gonna cut out all these introns. Okay, cut out all this intron. So in RNA splicing, you remove all the non-coding introns and you join together the exons. So in the end, we only have the exons. Do you understand? So it's like, yeah, go ahead.
So you keep all the exons, and then the introns, you just like get rid of them, like throw them out. You don't need them.
That's right, just delete them because they don't code for anything anyways. They don't have the instruction to make the proteins. They're just like filler DNA that we end up copying. Okay. It's like I don't know, think of like a magazine, right? If you want to copy magazine, there are some articles in it, and then you end up copying the not the commercials, what do you call it? The advertisements, right? We don't need the advertisement. We can just rip them out, remove them. They don't do anything. They don't have the instruction to make anything. Okay, they're just fillers. That's what the introns are. The introns are the advertisement. We don't need them. Got it? So you remove, you add the 5' cap, you add the 3' tails. Then you take away the junk DNA, the non-coding DNA, the introns, and you glue together the exons. The exons are the coding regions that are going to be expressed in the proteins.
Sorry, where did the purple line go?
Oh,
Like,
the
'cause that
purple,
was
no,
not
the
it.
purple, yeah, I should have used purple here to represent the stuff. This is the purple line is this thing. Yeah, I just use a different color. That's actually a good point. I probably should change them, but it won't let me anymore. Sorry. Okay, so this is the overview here, right? We did the transcription here, and then we have this pre-mRNA ribbon. If you pay attention to the color, you can see there is the darker pink, that's the exon, and then there is the lighter pink, which is the intron. So we're gonna remove all the light pink, and then it's just dark pink in the end. There's the 5' cap here on one end, and then there's the poly A tail. This thing's gonna go into the cytoplasm, and then we are going to use the information to make the protein. It will all come together, okay, in just a bit. Hopefully it'll all come together. Okay, right now it's still a little bit like unsure, like how does that make protein and so on and so forth, but hopefully it will make sense soon. Okay, let's just do a couple questions just to make sure that you are together with me with the terms and all that. The beginning of a gene. What do we call the beginning of the gene?
The promoter.
The promoter, that's correct. The promoter marks the beginning of a gene, and more importantly, it is rich in A and T. Lots of A and T's in it, make it easier to pull apart. Okay, and just to go through some of the other terms, initiation is the first step in transcription. Exons. Do we want to keep the exons, or do we want to throw them away? Keep them?
Keep.
That's right, we're gonna keep them because they're expressed in the end. They are the instruction. Introns, we remove them. Okay, and terminator is the end of a gene. Okay? So the gene is promoter, and then the core stuff in the middle, and then terminator. During post-transcriptional modification, what is being added to the 3' end? If you have been paying attention, you should know the answer is what are we adding?
The poly A tail, so adenine.
That's right, poly A tail. That's correct. Okay. Poly A tail. On the 5' end, that's the guanine. Okay. What is removed during post-transcriptional modification? Well, we just talked about that. Even in the last question. Hopefully, you know the answer is introns, right? Introns. All right. The next half an hour or so is gonna be important. If you have to learn what I teach you in the next 30 minutes in order for the rest of the lecture to make sense. Okay, like if you don't understand this next part, Thursday's class will absolutely be a disaster. Right, so please pay extra attention. Translation. Okay, translation. So quick review of the things that we already know. DNA and RNA are nucleotides. They are made of nucleotides, and in DNA is ACTG. In RNA, of course, is ACUG. So there are only 4 different nucleotides, right? For for proteins, the building block is amino acid. We learned that long time ago, and you should know that there are 20 amino acids. this is the chromosome, from one end to another, and then you have different section. The sections are the genes, right? And so we make a copy of the gene that we need to make the protein. So here in this example, we have a template DNA. So when we make the RNA, hopefully you know by now that the RNA is going to be complementary to the template. That's why if there's an A, when you build it, the RNA polymerase is going to put a U. When there's a C, you're going to put a G, and so on and so forth, right? It turns out every 3 nucleotide is called a codon. A codon. And each codon encodes for an amino acid. So UGG codes for the amino acid tryptophan. UUU codes for the amino acid phenylalanine, and so on and so forth. Okay? So I want you to think about this, and then tell me, how many codons can we create? Okay, remember, we have only 4 nucleotides, right? A will use the RNA. Okay, A, U, C, G. There are 4 nucleotides. And if I have 3 codes, 3 of the nucleotides, that makes up a codon. So I am asking you, how many codons exist? What's the maximum number of codons that we can create with 4 letters?
One, because you can't just have like G, for example, hanging out by itself, right?
Oh, but over here, look, I already have 4 different codons here, right? Like UGG is one codon, UUU is another codon, right? They are all different, right? So, how many more can I create? I can give you another one. I can have AAA, for example. I can have AUC, that's another codon, right? CCC, that's another codon. So I'm asking you, how many codons can we create in total? Okay, so someone says
then
6.
a whole bunch of them, because there's endless possibilities of combinations.
A whole bunch of them, I agree, but not endless though.
Okay.
Okay, yeah, a good try. Anyone else? Okay, let's have a math lesson, guys. Well, I have three slots here, right? 3 spots to fill. How many options do I have for the first spot, for the first blank? What are the options? Well, it could be a A, it could be a U, it could be a C, or it could be a G, right? So there are a total of 4 possibilities for the first blank. Do you agree? How many possibilities for the second blank? Well, it can be an A, it could be a U, it can be a C, or it can be a G. Also 4 possibilities. And then, what about the last blank? Well, you know how this goes. Also 4 possibilities. So if you have 4 choices for the first one, 4 choices for the second one, and 4 choices for the last one, it turns out the total number of possibilities, you just multiply them together, you get 64. Okay? It's like this. If you have 4 different hats, 4 different t-shirt, and 4 different pair of pants, how many different combinations you can have? Well, it's 4 times 4 times 4. Maybe that's a bad analogy. Okay, how about this? Let me just show you another one. I need a blank page here. Blank page. There we go. Okay. Let's say you roll a dice. Okay, roll a dice. How many outcome for rolling a dice? Rolling a dice. How many outcome, guys? Humor me. You know the answer, right? Don't you? 6, right? Okay, you can get 1, 2, 3, 4, 5, or 6. What about flip a coin? Flip a coin. How many outcome?
2.
2. That's right. So if you flip a coin and roll a dice at the same time, how many outcomes in total? Well, you can have a head or tail, and then 1, 2, 3, 4, 5. 6. And then for the tail, 1, 2, 3, 4, 5, 6, right? So it's just 6 times 2, which is 12 different outcomes. Do you understand? So if you have a bunch of different things happening and you want to know how many combinations are there, you just multiply all the options together. Okay? So if you have 4 choices for the first one, 4 choices for the second blank, and 4 choices for the last blank, there would be a total of 64 different codons that exist. Okay? However, we only have 20 amino acids, right? 20 amino acids. What does it mean? We have more, there are more codons than amino acid, which means there are some repeats. Okay, there are some repeats. Right? 2 different codes, 2 different codons can give you the same amino acid. Okay. So just like transcription, when we do translation, when we build proteins, we need to know where to start and where to end. So of the 64 codons, there is something called the start codon. The start codon is the beginning of your protein, and it's always AUG. You can remember August, the start of the school year or something, end of August, start of school year. Okay, so AUG, that's the start codon. And then what about stop? When are you going to stop? Well, the stop codon is going to be one of these 3 things: UGA, UGA, UAA, and UAG. And when you get to one of these things, you stop making the protein. Now, you always gonna have access to this chart. You should print one out for the test actually, because I don't think I've given it to you in the question, so you will have to print it out yourself to use it on the test. You might have it on your phone or something like that. Okay, this tells you how to get the code, and I'll teach you how to use the table. And you can see here, right, that's like the stop right here, and then that's the start codon is AUG. I told you, but then the stop codons are all written here, right? And you should memorize the stop codon because we need it for some other concept that we will learn, and it's like takes too long for you to look them up on the table. So I need you to memorize the stop codon. This is how you can remember it, okay? So UGA, you can think of it like this, like you're taking the biology course, and then you're like, okay, you got A in biology, you are awesome. Okay, you are good. Okay, so that's how you can remember it. Okay, you got A, UGA, you are awesome, UAA, you are good. So those are the 3 stop codon. Okay, start codon, AUG, stop codon, one of these 3. Are we together for now? Okay. So this is called the codon chart, codon table. Like I said, print one out for the test, have it on your phone. Don't have it on your computer though, because you have the lockdown browser, right? So you won't be able to open it if it's on your computer. So how do we use it? Let's say we have a strand of messenger RNA here, strand of messenger RNA. This strand has gone through the post-transcriptional modification. Okay, you removed the introns already, and now it's just the coding information. So how do we convert it into protein? Right, so the ribosome will come, and then it will read the code 3 at a time. So AUG, that's the start codon. Okay? So what does it code for? So this is the this column here. This is the first nucleotide. This is the second nucleotide, and this is the third nucleotide. So this is AUG. So I look for A here. A is right here for the first column. I look for A. That's A. And then I look for U in the second one here. So U is here. Okay. I mean, you don't have to draw on it all the time like this. I'm just showing you, right? So where they intersect is where I need to look. Okay, so A and U intersects right here in this box. And then I'm gonna need to look for G, which is, I look for the last letter here. Okay, so G is right here. So that means the amino acid is MET. So AUG codes for MET. And since AUG is the start codon, all your protein starts with MET. Let's do another one. Okay, watch this. And I hope you have this chart with you, right? Otherwise, it's very difficult for you to follow. Next one we're looking for is AUU. Okay. So A is here, U is here. So that means I'm looking at this box here, right? And then I look for U. So that's right? AUU is ILE. Let's do one more. CCU. CCU. So C, so we're here. And then U. That's pro, right? Proline. And all these are amino acids. And you can see their redundancy, right? Look, CCU is proline, CCC is proline, CCA, CCG, they're all proline. Why do we have redundancy? Because we have more codons, 64 of them, than amino acid, 20. Okay, so can we please finish the rest of it? All right, I'll give you a moment. And if you still don't know how to use the chart, just send me a message, okay? You can private message me if you don't want to tell the whole class, or you can tell the whole class, and then I can go through it again. Because you need to do this in order to understand the rest of the lecture. Okay, yeah. CAU. That's histidine, HIS. CGC is arginine, ARG. You don't need to know the full name. I just feel like saying them. UAU is tyrosine. UUC and UUU, PHE, and then UAA. Remember, you got A, you are good, you're awesome. Those are the stop codons. Then we just stop. That's it. Does anybody have any question? Okay. I need all of you to do this poll question, if possible. Okay. So that I know all of you can use the chart. Okay. Please.
Sir, can you go back to the chart and then screenshot it?
Oh, sure. Okay. CGA. Okay, we're looking for CGA. Yeah. So like I said, right, you guys gotta print your own chart. for the test, please. Okay, CGA is the question. You can find the thing first, and then I'll show you the choices again. CGA. Okay, so what is the answer? Choose the answer. Okay, anyone else? Yeah, I got 50%. Are you there? And then some of you type it in the chat box as well. Okay. Are we good? We understand? Why is ARG? Don't be shy. If you don't understand, I can show you again. Because if you don't know how to use the chart, we are gonna be in some trouble for the rest of the lecture. Not the rest of today, but like the rest of lecture 7. That's what I mean. Okay, fantastic. This next one is challenging. Okay, maybe we just do it together. What? Oh, sorry. Which of the following? Which of the following is a stop codon? Is a stop codon on the template DNA? Template DNA. Okay, so let's write down the stop codons on the messenger RNA first. Okay, do you remember what they are? On the messenger RNA. Okay.
UAA,
Yeah.
UAG, U.
Yep, almost there.
UGA.
That's right. You got A, right? Okay, so you got A. You're awesome. You're good. Okay, cheesy as it is, people tell me this is the only thing that they remember by the end of the course. Okay, so UGA, UAA, UAG. Now listen, right? This is messenger RNA. The question is not asking you about messenger RNA. It's asking you what it looks like on that template DNA. Okay, so that's why it's a difficult question. We got to convert back. All right, so like, coding DNA and template DNA. Now, if you have that cheat sheet that I made you on the sticky tag, you can take a look. The relationship between mRNA and coding is that they are the same, but instead of U, we will have T. So TAA, right? TAA, TAG, TGA. It's the same, but replace the A, the T with a U. And then we got to go one more step further. We want the template DNA. So relationship between template and coding again is complementary. Okay, so T is going to be A, ATT, ATC, and ACT. Okay, so I look for these ones. ACT, ATT. Those are the only one that shows up. Okay. Do you understand? It's not E, right? It's not E because we're looking for the stop codon on the template DNA. Remember, even though the table that we use is mRNA, ultimately these things came from the DNA, right? Ultimately came from the coding DNA. Right? Give this a little bit of time to think about it. I'm gonna do one more question with you all, and then that will be it for today. And we'll come back for more on Thursday. Okay, so here we go. Decode the following messenger RNA sequence into amino acid. Oh, actually, you know what? You probably can do this yourself. I actually want to do this one with you. Example 2. Okay, example 2. Example 1 is easy, right? Like you just look it up and practice using the chart. All right. Let's do example 2. we have the template strand now. So when you have a template strand, we have to convert it into messenger RNA first. Right? We have to do the transcription first. Okay, so TAC, what should I write here? I'll give you a moment to think about it. Okay, like look at the cheat sheet.
UTG,
What is it again? Sorry.
UTG,
UTG. Okay. Anyone else? Remember, we are looking for the messenger RNA first. Okay, we're not changing it to protein yet. Okay. Okay. A lot of you telling me is AUG. Okay. Did you like go all the way, guys? Like, did you do like an extra step already? Is that what you did? Roy, Oge, and Kiria. Is that what you guys did? Did you go all the way to the protein? Okay. Okay, great. So we're not going to do the proteins yet. I just want to make sure that we get the mRNA first. If you're confident, keep going. But the answer is actually, it's actually AUG here. Remember, this is the template. The relationship between the template and the messenger RNA is that they are complementary. Okay. If I give you the coding, then you can copy it as is. Okay, so this is complementary. I usually put a little dash here so it's not too crowded. Visually, it's easier for me to see. Okay, next one. GTA is gonna be CAT. CAU, I'm sorry. There's no diamine. And then GGA, UUA, and then UGU, CCG, UGA. Okay. What's the relationship again? Complementary. Then what are we gonna do? Then we look it up, right? AUG. So AUG, it's MET. It's always MET for the first one, right? Well, I mean, like when I give you a sequence on a test, it might not necessarily be the beginning of the protein. It might be like a chunk of it in the middle. So you know, don't think you're wrong if your first one does not work out to be the MET. Okay, what about the GT? The CAU. 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 It's gonna be HIS. Okay, CAU, right? Okay, Sarah, is that okay? You got it? CAU, yeah. And then the next one, GGA. GGA. UUA. UGU. Okay. And then CCG. CCG is pro. And then you got a UGA is stop. Did I miss anything? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. There it is. So that's what happens in the body. If you need to make this protein, you need to find the gene in your DNA. You got to copy it in the form of a messenger RNA. And then your ribosome will read the codons and build the amino acids. Connect the amino acids, which then becomes the protein. Now, there is one key assumption that we've made here in this exercise. You probably didn't think about it, but what we are assuming here is we're gonna assume this section is all exons, right? Okay. But in reality, the DNA strand is much longer. So the mRNA is much longer, and then you actually have to remove the junk first, right? The intron. Okay? But in all the questions I will give you, you can just assume it is all exon. It's all just gonna get converted into proteins in the end. All right. Now we did not get to the study guide, but you know, fear not. we're gonna go back to it for sure next class, right? But there are these questions. I mean, I haven't teach you the anti-codon yet. I will, but for now, just practice using the table. There are a few other questions to try. Example 3 here. And then example 1, redo some of the ones that we've done. Just get really comfortable with the table. Definitely print a big one for next class. We will be using it extensively for the rest of the lecture. Okay. Good luck, I guess. Right. Go through the material. If you're confused, email me. Otherwise, I will see you all on Thursday. Okay. Thank you for coming. Have a good rest of your day.
Thank
Bye-bye.
you, sir.
Yep.
Thank you, sir. All the best.
Okay. Thank you.